Hi, I'm Jen Payne. I'm from Monash University as part of the BDI and EMBL Australia, and I'm part of Max Kryle's group where we work on antibiotic-resistant superbugs. And I wanted to tell you a bit about the work that I did in the US last year, thanks to two fellowships, one from VESCI, the Victorian Fellowship, and the Hugh Rogers Fellowship that I received from Melbourne and Boston Sister City Association. And they allowed me to do some networking, some great science, and form some fantastic collaborations. And that was all part and thanks to Daniel Arenia and Felix Elliott at the Massachusetts General Hospital and the Harvard Medical School. So I spent over 6 months last year in Boston in two different stints, and over there I was able to do some fantastic research where we looked at immune cell migration towards infections, and we did this on a single cell level. And we were able to do this because I was visiting our collaborators that are experts in this technique of microfluidics, and we can create little mazes basically for immune cells to migrate through. And then so picture the immune cells as mice, and your typical kind of science experiment where you put the mice in a maze and they're trying to get to cheese, but instead of cheese we've got the bacterial infection. And so I'm looking at how our antibiotics influence the mice getting to the cheese, so the immune cells getting to the bacteria. And so we were able to look at that and how our antibiotics actually improve the immune cell's ability to track down and find the bacteria through this maze. And the bacteria that we were looking at were the Staphylococcus aureus or the MRSA, these superbugs, these supervillains. And we have some great leads for being able to treat these supervillains for the future. What we also were able to look at in the second visit that I did was neutrophil swarming, which is the process of the innate immune cells known as neutrophils, and they move towards the site of infection. They basically create a giant dog pile on top of the bacteria to stop it from escaping, and then they call in the cavalry and try and get more and more cells to come in and help fight this infection. So you've basically got this pile of immune cells piling up on top of the infection, and that's neutrophil swarming. And so we haven't in the past really been able to research this process. So we've been able to do it in the body, but we haven't really been able to look at how drugs affect this. And so due to being able to print onto slides, and we can print bacteria onto slides and then print them into a grid and then look at the migration of the immune cells towards these, and we can see that our drugs affect the swarming process and speed up the swarming so that the infection is cleared. So it looks like we have some new great targets for antibiotics that are improving the immune response to bacterial infections that would otherwise hide away in our tissues and develop into deadly diseases.